Alright, so I'll be going through figure 1, where they are establishing that Draxin is expressed in the pre-migratory cranial neuronal crest, and then is downregulated as the cells undergo this epithelial-mesenchymal transition. And so overall, they're establishing the Draxin expression pattern as the cell is, or as the embryo is developing. And so a little bit of overview and background. They are using whole-mounted in situ hybridization, which is a technique used to visualize the location of specific expressed DNA and RNA sequences in embryos. And this is done by synthesizing a probe that is specific to the sequence and will integrate into either the DNA or RNA, which will then be expressed and will help us visualize the location of that sequence that was expressed. They also use whole mount immunohistochemistry, where they incubate the tissues with their primary species-specific antibodies, as well as their secondary antibodies that are labeled with Alexa fluorophores. And the model they used in this figure was the chicken embryo, which can be incubated and grown to specific Hamburger-Hamilton stages. And these Hamburger-Hamilton stages are important because they're well-characterized chick embryo developmental stages where the specific structures and sort of the stage of the neuronal tube is known and can be used to track the expression of our Draxin. And beginning here, we look at these chromogenic in situ hybridization experiments, where the goal was to determine the expression pattern of Draxin. And so that's the general expression pattern of Draxin and where it's expressed and what part of the neuronal tube and how long is it there, those sort of things. And so they use a digoxygenin labeled antisense RNA probe to probe for Draxin, which was then visualized here. And it was determined that the first visible expression of Draxin happens in the Hamburger-Hamilton stage 9, which is pretty early on in the developmental stages. And so we see that at the early stage of HH9, the expression of Draxin is happening sort of in the head of the neuronal tube. As we move into the intermediate stage, the Draxin is not as intensely visualized in the head, but has now progressed downward towards the tail. And in the late stage of HH9, we see that it looks sort of like it has diffused out into the surrounding tissues and is now mainly concentrated in the tail. And so if this were a dynamic video, the likelihood of Draxin expression would be that the intensities of Draxin is sort of moving downward like a wave or like it's being propagated and removed from the head area, which is something we know happens in the cranial neuronal crest development. And so they also perform these cryosectioning images here, where the tissue was prepped and frozen and then transversely cut, and the cuts occurred at these dotted lines here, labeled as their primes. And so in A prime, the concentration of Draxin is shown in this light blue color, and it's at the tip of the neuronal tube. Next, we look at the intermediate Hamburger-Hamilton stage, and the intensity sort of reaches a peak when compared to the final or the late stage here, where the Draxin is sort of moved out of the neuronal tube, and we now only see the neuronal tube tissue. And so moving forward, after determining the Draxin expression, the general Draxin expression, they wanted to see the pattern of expression relative to the actual neuronal crest development. And they did this by tracking two developmental markers, PAX7 and HNK1, where PAX7 is this sort of early stage marker that is expressed during the pre-migratory events, and HNK1, which is a mesenchymal marker. And from the name alone, we can assume that it is expressed in the late stages or during this migratory cell stage where the cells have completed their epithelial to mesenchymal transition. And so these are fluorescence imaging or fluorescence visualization images where they used immunostaining to stain PAX7 as well as HNK1. And we see that PAX7 is pseudo-colored in green and HNK1 is shown in purple. On the other hand here, we see the second embryo where they used the fluorescence in situ hybridization technique to visualize Draxin, and then they immunostained again HNK1 here in purple. And so we begin with figure D at the early stage of this HH9, where we see that PAX7 is expressed alone. Moving into this intermediate stage, we see the presence of HNK1 beginning at the head of the embryo, and then towards the late stages of HH9, we see that the HNK1 has sort of increased in concentration and also progressed sort of downwards towards the tail. Now, in the stage-matched embryos expressing Draxin, it's assumed that the stages are relatively at the same time. However, this is a different embryo, and the assumption here is that if you were to overlay these, we'd see that Draxin is present at the same time as PAX7, and so the expression of these two happen or are overlapped, meaning that Draxin is more than likely expressed during the early stages of the neuronal crest development. And so moving forward into this intermediate stage, even though we see that HNK1 is present in this embryo, there was no HNK1 markers visible for our intermediate stage with Draxin. However, at the late stage, we see the sort of similar pattern of HNK1 expression, and then we see that Draxin is sort of no longer present, as shown by like the relative decrease in intensity. And so what this is demonstrating is that Draxin is co-localized with our PAX7 marker, meaning that these two are both expressed during the pre-migratory stage of the neuronal crest development. But then as our mesenchymal cells begin to appear, the expression of Draxin sort of decreases and is inhibited in a way by the presence of these mesenchymal cells, which are indicative of this migratory stage in the neuronal crest development. And so some of the conclusions they drew from this is that Draxin is present in the early stages of neuronal crest development, and that as the development progresses and the cell begins to undergo this epithelial to mesenchymal transition, Draxin is visibly downregulated. And so they are establishing that there is a relationship between Draxin and the WNT signaling pathway, which is known to be responsible or to play a role in modulating neuronal crest development. And so this visualizing or imaging experiment here sort of set the groundwork or sort of set the background in establishing the role that Draxin can potentially play, or the fact that Draxin is involved in this signaling pathway. Sorry for all of that information that I just mumbled through.